So on to our last two sections of the lymphatic system. Section 19.5 is called MALT, and then we'll talk briefly about the thymus. MALT stands for mucosa. Okay, that's a big part of this, associated lymphoid tissue. So these are lymphoid tissues in mucous membranes throughout the body. So like when we look at Peyer's patches, you're going to have the serosa or adventitia layer, then you'll have a muscular layer, then you've got that submucosa and the mucosa. So for MALT, it is close to the lumen of these hollow organs, if it's a hollow organ. Mucosa-associated lymphoid tissue, so MALT, protects from pathogens trying to enter the body. And again, if you look at, if you think of the GI tract as being an extension of the external environment, by having MALT tissue here, it's going to keep bacteria from getting outside of that GI tract and into your internal cavity. So a lot of the MALT tissues are all about containment. The tonsils are a little bit different in that they are kind of an early detection system because they're in that oral cavity. And so your body would rather know about a pathogen sooner rather than later. So it will actually capture pathogens that are coming into your body and intentionally capture them in order to alert your body to pick it up and, produce some antibodies, get some T cells out there, get ready for what's coming. Hopefully that pathogen will get digested and not become a problem. But just in case it is something that is going to bypass your defenses, your body gets a little bit of a head start. So we'll talk about the largest collections of MALT separately, the tonsils, the Peyer patches, and the appendix. But these are found just here, there, and everywhere in the mucosa of the respiratory tract, because that makes sense. So we breathe in potential pathogens, some of the genitourinary organs, and then the digestive tract is mainly associated with Peyer's patches in the appendix. Okay. So the tonsils, there are actually 4 sets of tonsils. A lot of times we think of there just being 3 sets. When sometimes we're talking about anatomy, we'll just talk about 3 sets, and we don't really talk about the tubal tonsils. But there are 4 sets of tonsils. There are the palatine tonsils at the posterior end of the oral cavity. So if you've had a child or you were a child that got a lot of strep throat infections and had your tonsils removed because you had 3 strep throat infections, I think in like 6 or 8 months, I don't know, it's a lot. These are the tonsils that get removed. So tonsils intentionally have what are called crypts, which are unencapsulated spaces that trap what's coming in. But some people have highly folded, invaginated crypts that then get infected rather than it, so they get overexposed and they get infected. So those are usually the individuals that end up having theirs removed because they're more problematic than helpful. And then they become a breeding area for that strep bacteria. The lingual tonsils are a collection of follicles at the base of the tongue. The pharyngeal tonsil, sometimes also called the adenoids, these are located in the posterior wall of the nasopharynx. And sometimes these pharyngeal tonsils will also get removed. Some individuals have very large pharyngeal tonsils and they cause problems with sleep apnea or snoring. And so then those get removed, but they're not usually because they're infected. They're just usually because they're too big and obstructing airflow. The tubal tonsils are the surroundings of the auditory tubes as they drain into the pharynx. Okay. So the tonsils' function is to gather and remove pathogens in food or air. They are, like I said, this first, like an early detection system. They contain follicles with germinal centers and scattered lymphocytes. So you'll get B lymphocytes and T lymphocytes. They are not fully encapsulated because they actually want to be exposed to whatever they're trapping. The overlying epithelium invaginates, it folds, forming these tonsillar crypts to trap bacteria. And if they're too extensive, that's when they start to get infected. Bacteria or particulate matter enter the crypts where they are trapped and destroyed. It's risky to lure bacteria into tissues, but allows the immune cells to become activated and build memory cells against these potential pathogens. The next two, Peyer's patches and the appendix, are associated with the intestinal tract. So Peyer's patches are going to be clusters of lymphoid follicles along the small intestine. And they are structurally similar to tonsils. They are going to destroy bacteria and generate memory lymphocytes. So I'm going to go to, this is I think a really nice histological section of either the jejunum or the ileum, where you have, your outer serosal layer, you have your outer muscular layer, then you get into submucosa and mucosa. And these are Peyer's patches in here. And one of their functions are these memory T lymphocytes. And memory lymphocytes, they get exposed to an antigen, and kind of like a plasma cell or antibody producing cell, they have this memory. They remember what they've seen before. And in B lymphocytes, it means that they'll start secreting antibodies against the antigen. But in memory T cells, they're like, oh, I've met you before, and I determined that you were non-self last time, or antigen, something needs to be destroyed. And it will just automatically attack it. It doesn't need that antibody tag or the dendritic cell tag to say this shouldn't be here. It remembers, and it will just automatically destroy it. And this is important in the intestinal tract because we do have populations of bacteria, especially when we get to the appendix and large intestine, lots of bacteria in there. Shouldn't be as much bacteria in the small intestine because we should keep it in the large intestine. But, we're not always 100% here. And so the Peyer's patches can identify a bacteria that shouldn't be there. Maybe it got through that ileocecal valve, or maybe it was in the feces, who knows. And it will then attack that bacteria and kill it off and prevent it from moving further up the digestive tract or outside the digestive tract. Okay. The appendix. So we went from Peyer's patches with the small intestine to the appendix and the large intestine. The appendix is just off of, so there's the cecum and then the ascending colon, transverse descending. So the appendix is about this long off of the cecum. It contains a large number of lymphoid follicles. It acts like Peyer patches. It's just not called Peyer patches because it's in this large intestine instead of the small intestine. Again, destroying bacteria and preventing them from breaching into the small intestine or breaching the intestinal wall. And these also work, there are other mechanisms, but they rely heavily on memory lymphocytes because it tends to be the same type of bacteria that they're encountering over and over again. On to section 19.6, the thymus. So the thymus is a bilobed lymphoid organ found in the inferior of the neck, and it extends into the mediastinum, can partially overlie the heart in a small child. So It's much larger in childhood, and then it starts to atrophy as an adult. Okay, so most active and largest in size during childhood. This is where T lymphocytes go to mature. So both B lymphocytes and T lymphocytes are produced in the bone marrow. Then the B lymphocytes stay there, but the T lymphocytes migrate out to the thymus, and they can spend months to years maturing. It stops growing during adolescence and then gradually atrophies. It still produces immunocompetent cells, although at a slower rate. Immune function does decline with age. The thymus is broken into lobules that contain a cortex and a medulla, just like your lymph nodes. Lymph nodes have cortex and medulla. Spleen has white pulp and red pulp. Kind of keep that in mind. Cortex is always pretty much where you're going to have the most divisions, where like you'll have follicles, and the thymus isn't any different. Cortex contains rapidly dividing lymphocytes, which make up the bulk of the thymic cells, and then some scattered macrophages. But the thymus is the home of T cells, and the medulla contains fewer lymphocytes and corpuscles. These thymic corpuscles are where regulatory T cells develop. Regulatory T cells actually suppress an immune response. So they find inflammation at a self tissue, so your own body tissue, and they sense that this immune response is occurring at your own tissue, and so then it secretes cytokines to suppress that inflammatory response. It's actually kind of sometimes it can be, they antagonize certain like cancer treatments, and so sometimes they will try and suppress the regulatory T cells. But in general, we want regulatory T cells, or else we would have an increase in autoimmune diseases or just bouts of autoimmune where we're attacking our own cells. Okay, so cortex, rapidly dividing lymphocytes, medulla, fewer lymphocytes, and the type of cell that is going to be found in the thymus are T lymphocytes. Thymus differs from other lymphoid organs in a few important ways. It has no true germination follicles, so it doesn't have any stem cell tissues, and it will lack B cells. It does not directly fight antigens. It's not like the spleen or your lymph nodes or MALT that have macrophages that are going to, and natural killer cells. It's not its role. Okay, so there might be a few cells that get destroyed here and there, but it's not its main function. Its function is T lymphocyte maturation. It also maintains a blood thymus barrier to keep immature T lymphocytes isolated to prevent premature activation. So thymus, it's not going to have germinal cells. It's not going to have B lymphocytes. It is there for T lymphocyte maturation. Another, and a very, I think it's the only one that is like this, the stroma is made up of epithelial cells, not reticular fibers. We've talked a lot about reticular fibers and how the majority of lymphatic organs are composed of reticular connective tissue. It's kind of a defining feature of the lymphatic tissues and organs. And the thymus is the one exception. So these are thymic corpuscles instead of follicles. This is where T cells are going to mature. They're also going to, majority of maturation actually occurs though here in the cortex. So thymus, T lymphocytes to where they mature, much more active in childhood, then function and size decreases from adolescence through adulthood. And that is really that's kind of the end. This is just where we've been. And to summarize, if you see an exam question that says like lymphatic tissue is reticular connective tissue, that's true. We kind of exclude the thymus when we're talking about that. So the vast majority of lymphoid organs and tissues have this reticular connective tissue. So I try very hard not to do trick questions. So try not to think about the one exception, like the lone macrophage that might be eating something. Think what happens 98, 99% of the time. Lymph nodes have a cortex and a medulla. The afferent vessels come in on this convex side, and then the efferent vessels exit at the hilum. The spleen is not divided into cortex and medulla. It's divided into red pulp and white pulp because it has two functions. The red pulp is going to be for storing platelets. This should be monocytes and iron. And then the white pulp is for lymphocyte activation and proliferation. So spleen has this dual personality with red pulp and white pulp. And we talked about malt with the tonsils and Peyer's patches and appendix, and it's really to prevent pathogens from penetrating that mucous membrane. And again, it doesn't have a cortex and a medulla. It's just arranged into these crypts instead. So for those of you who joined us a few minutes late, the exam is a single exam for reproductive and lymphatics because there are only 17 questions from the lymphatic system, and half of those are multiple choice with just one answer. Like it doesn't say select all of these that could apply. It says choose the correct one or choose the incorrect one. So those are a little bit quicker. So that's all I have. If you guys have questions about the course or anything, I'm happy to answer those as well.